video, let us talk about the concept of cell cycle. So let's begin. So first of all, the idea that we have to understand is that cell cycle is a cycle, right? You may feel it to be very dumb, or it's the life cycle of a cell. So it's life cycle of a cell, and the cell cycle that we are going to study, it's of somatic cells, right? Somatic cells, that means the cells that undergo mitosis. The cell cycle of cells which undergo meiosis, or what you call as meiosis, is not in the part of our syllabus, so we are not going to discuss about that. Now this cell cycle is divided into two parts. It has a mitotic part and it has a mitotic phase and interphase. So it has a mitotic phase where it undergoes mitosis, and it has a interphase, right? Now this interphase has different divisions. It has G1, it has S, and it has G2 phases. So the interphase is known as the resting phase of the cell. Resting phase of the cell, which is a misnomer. So you have to remember, interphase is one of the most active phases of cell. When the cell grows in size, it does protein synthesis, it does DNA replication, it does duplication of organelles, it does RNA synthesis, and a hell lot of stuff. But why it is known as resting phase? Because it is not concerned, it is resting in terms of cell division. Resting in terms of cell division. As no cell division is going on, it is known as the resting phase. So it is a misnomer that you have to keep in mind. But interphase is in fact the most active phase of the cell cycle. So interphase is the most active phase. Most active phase of cell cycle is your interphase. I hope you've got it, right? So with this basic concept about cell cycle, let's delve into deep of the cell cycle. So let's start. So now cell cycle starts with a phase known as G1 phase, which is also known as the growth 1 phase. So G1 stands for growth 1. Growth 1 phase. So it is known as growth 1 phase because this is the phase in which the cell starts growing in size and in its content for the very first time after mitosis. So the interphase starts with growth 1. Now in growth 1 phase, three very important thing happens. First of all, organelle duplication, organelle duplication, most of the organelle undergo duplication, and secondly, the most important thing is that synthesis of RNA, DNTPs, that is nucleotides, and proteins take place. Synthesis take place. So proteins are for the growth of the cell and growth in size. As you can understand that if the organelles duplicate, so they need more space to live, so they need growth in size of the cell, and for the growth in size of the cell you need a lot of proteins to make to produce the cytoplasm or the protoplasm, that is in which the organelles are. So for that there's protein synthesis, and for the upcoming DNA duplication that is going to be in the later phases of cell cycle, you need to prepare RNA and DNTPs and the enzymes, which are also proteins of DNA replication, right? And from RNAs the proteins are produced, right? So this is the in crux, the idea of G1 phase. After G1 phase, we move into the next phase of cell cycle, that is the S phase. So S phase is concerned with mainly one thing, one very important thing, that is DNA replication. DNA replication. DNA replication is the only major event that occurs in S phase, and this S in this S phase stands for synthesis, which refers to synthesis of DNA. So this is the idea of S phase. And another very important thing about S phase is that you have to keep in mind the amount of DNA. As the DNA is undergoing duplication, so if the amount of DNA is 2C at the beginning of S phase, the amount of DNA becomes 4C at the end of S phase. The amount of DNA just doubles. This process is just duplication of DNA, just DNA gets doubled, right? So this is the main idea of S phase. So after S phase, the cell goes into the growth phase 2 or the G2 phase. So G stands for growth phase 2. G stands for growth 2, growth 2. So what happens in G2 phase? That is very important. So in G2 phase, the mitochondria, plastid, that is the chloroplast, chromoplast, leucoplast, all those plastids, and your Golgi body duplicate. Right, this is very important, that mitochondria, plastid, Golgi body duplicate, and this phase tubulin protein synthesis takes place. Tubulin protein synthesis takes place. Now this tubulin protein creates the spindle machinery of the cell cycle. So tubulin protein is very important, and with this preparation, so after G2 phase, cell has grown in size, it has enough cytoplasm to divide into two cells, it has a lot of proteins, it has double number of organelles, it has double number of DNA, it has tubulin protein for the tubulin protein, it has mitochondria, plastid, Golgi body, all in double number to be divided into two cells. And with all this preparation, it gets into the mitotic phase. Now this mitotic phase is again divided into two phases that you know. One is the karyokinesis, another one is the cytokinesis. First it starts, okay, so over here there's a step of karyokinesis, sorry, yeah, black pen, so right, so karyokinesis, then there's part of cytokinesis, that is the cytoplasm division, and karyokinesis is the nucleus division. In nuclear division we have four stages: prophase, metaphase, anaphase, and telophase, and then we have the cytokinesis, right? But we will be dealing in details with the mitotic phase when we will be dealing with a dedicated video of mitosis. In that we will discuss in details each of these phases and cytokinesis in details, but for now we are just leaving it over here. Now one important point that I skipped in G1 phase is that the length is variable. The length of G1 phase is variable. So in cells which grow more in size, I mean they undergo frequent divisions, they show smaller G1 phases, and the cells which do not undergo frequent divisions, they show longer G1 phases. So let's bring in an analogy over here to understand this concept. Suppose you have exams every consecutive day, that is from Monday, then Tuesday, then Wednesday, then Thursday, Friday, all the way till Saturday, right? So then your exam preparation on every day will be just the evening and night before the examination. But if you have exams every week, all these 6 exams are divided into a span of 6 weeks, and you get exam every Monday or every Tuesday, then your preparatory phase becomes almost a week, from Monday to next Monday. Alright, so this means if the frequency of exam is increased, your preparatory phase is decreased, and if your frequency of exam is decreased, your preparatory phase increases. Similarly, if frequency of division increases, then length of G1 phase decreases, and if the frequency of division increases, frequency of, sorry I got messed up, the frequency of division if increase, G1 phase will decrease, length of G1 phase, and if frequency of division decrease, that means there's infrequent divisions, then G1 phase will increase in length, right? Okay, so one last thing before we finish it off, that is the G0 phase. So what is G0 phase? G0 phase is nothing but an escape from the cell cycle. So there are certain cells which are well differentiated in our body, and they do not require frequent divisions. So and there are other cells which do not undergo divisions at all. Those cells escape the cell cycle and go and rest in the G0 phase. So certain cells, like a very good example that you have is your neurons or nerve cells, they rest in the G0 phase, they don't divide. So this G0 phase you have to keep in mind. G0 phase is nothing but it is an escape from the cell cycle. And another very important point before we finish it off, that is the regulation of cell cycle, the regulation of cell cycle, right? What regulates the cell cycle? So there are two components which regulate the cell cycle. One is cyclins and cyclin dependent kinases, right? So what are kinases? Kinases are certain enzymes, right? It's an enzyme. Now it's that substrate. What kinases does, if there's a substrate, the kinases phosphorylate the substrate by using what? By using ATP. They break down ATP into ADP and use a phosphate to phosphorylate the substrate. So kinases are enzymes which are related with the phosphorylation of substrates using ATP. So the regulation of cell cycle is done by cyclins and cyclin dependent kinases. So what is cyclin? Cyclin is a protein, and cyclin dependent kinase is an enzyme which is again a protein which deals with the active phosphorylation or phosphorylation of substrates using ATP. So this was the important point about cell cycle. So this was in brief the discussion of cell cycle of somatic cells.